now we can talk about how we actually perform the accelerant analysis. So once we collect all that evidence, what do we do with it? So gas chromatography is going to be ideally suited for this. First of all, there's probably a combination of different substances in there. And gas chromatography, you may recall from when we did our drugs chapter and a little bit in the paint chapter. But you'll recall that it's a separation technique, and it separates different gases based off of or liquids or gases based off of their boiling points. And so we see that this works particularly well because these accelerants are often in gas form. You know, they're liquids that evaporate easily and are in gas form. So this is perfectly set up. It's pretty sensitive, it's reliable. Of course, if we pair it with mass spectroscopy, remember we talked about GCMS, this is a perfect technique. We can detect small differences in different petroleum-based accelerants like gasoline and butane using gas chromatography. So we are going to, as we just reminding you, it separates by boiling point, and we get a bunch of peaks based off of retention time. The low boiling point accelerants would come out first, and the high boiling point would come out last. And then we can do comparison. In this case, there are reference files that will sort of say, okay, this is what this particular gasoline blend looks like, this is what this lighter fluid looks like, this is what this engine cleaner looks like. All of these things could be serving as accelerants, and so there are databases of showing like this is what you would get from a gas chromatography column under certain conditions, so that that can be used as a good comparison source. Of course, you'd really want to have two samples side by side run in the same machine under the same conditions for best results. Again, we're going to compare that pattern generated from, so the sample comes in. We do either the headspace technique or that vapor concentration technique to sort of collect the sample from the fire debris, and then it's going to be run through the GC and then compared to this library of different setups. So here's what it might look like. Again, remember, there's a whole lot of stuff in here. And in fact, even if you get gasoline, it's a blend of lots of different products. So some, there's different blends of octane and decane. So there's different kinds of materials in there. There's also additives in there, and they might be incomplete. So you have a lot of different peaks because there's a lot of different substances in there. So they're fairly complex. But as you can see, in this case, we can see great overlap of these peaks, and the pattern looks pretty similar between these two substances, suggesting that this top substance might be a mixture that contains this bottom substance in it. And from this, then, further work could be done to kind of confirm on that. And this is, again, a gas chromatography machine. I'm just reminding you, this is kind of a review from drugs and the other one. So we see an unknown. Right, here would be our unknown, here's our known gasoline, and then we could actually run it through, once we take that unknown, run it through after GC through the mass spectrometer, and we can actually confirm exactly pretty much what it is.